Hello everyone, let's discuss about vitamin B6 or pyridoxine. This is a water soluble vitamin. Synonym is rat antidermatitis factor. Rat antidermatitis factor. This is the structure of pyridoxine. It is also called as Pyridoxol. Then it is other name of pyridoxine is Adermin. Adermin. Then it is chemically 2-methyl 3-hydroxy 4,5 dihydroxymethyl pyridine. It is 2-methyl 3-hydroxy 4,5 dihydroxymethyl pyridine. It occurs in association with aldehyde pyridoxal. This is the aldehyde form. Here CHO group. In place of CH2OH there is CHO group. It is called as pyridoxal. So it occurs in association with the aldehyde form pyridoxal and an amine form pyridoxamine. Here CH2NH2. All three forms exhibit vitamin B6 activity: pyridoxine, pyridoxal and pyridoxamine. The biological active forms are forms of vitamin B6. Pyridoxal phosphate and pyridoxamine phosphate are the biological active forms of vitamin B6. Pyridoxal phosphate and pyridoxamine phosphate. So here it is changed to OP. Here also one phosphate group. This is the biological active forms: pyridoxal phosphate and pyridoxamine phosphate. That means active forms are the phosphorylated derivatives. Phosphorylation involves a hydroxymethyl group CH2OH at the 5th position. This CH2OH is replaced by this phosphorylation is replaced by a phosphate group. So it is in the 5th position in pyridine ring. These forms occur in nature largely in combination with protein or apoenzyme. Next we will see the formation of pyridoxal phosphate. Formation of this pyridoxal phosphate. This is the biologically active form. Pyridoxal phosphate. Phosphorylation takes place in liver, brain and other tissues with the help of ATP, Zn2 plus and enzyme pyridoxal kinase. Pyridoxal kinase, ATP, Zn plus plus. Two pathways are there. In first, pyridoxine. In first pathway, pyridoxine is first converted to pyridoxal. Pyridoxal by the enzyme dehydrogenase. By the enzyme dehydrogenase, pyridoxine converted to pyridoxal. Here FPH2. Then by the enzyme kinase, pyridoxal is converted to pyridoxal phosphate. Pyridoxal phosphate. Here one ATP, ADP plus PI. This is the first pathway. So in second pathway, pyridoxine first undergoes phosphorylation with the help of ATP dependent kinase. Pyridoxine first undergoes phosphorylation with the help of ATP dependent kinase to produce pyridoxal phosphate. This is the formation of pyridoxal phosphate. Next we will see biosynthesis of vitamin B6. Biosynthesis of vitamin B6. Vitamin B6 can be formed by microorganisms and also by plants. The human beings cannot synthesize the vitamin, hence it has to be supplied by the diet. Intestinal bacteria can synthesize the vitamin. Next is metabolism. First one is absorption. This vitamin B6, dietary vitamin B6 is readily absorbed by small intestine. Coming to excretion, pyridoxal and pyridoxamine are excreted in urine. Pyridoxal and pyridoxamine are excreted in urine in small amounts. that is about 0.5 to 0.7 mg daily. Pyridoxal and Pyridoxamine, these are excreted in urine in about 0.5 to 0.7 mg daily. Major urinary metabolite is an inactive form of pyridoxine, that is 4-pyridoxic acid. 4-pyridoxic acid. This is the major urinary metabolite, but it is inactive form. 4-pyridoxic acid. So here one COH group. This is the structure of 4-pyridoxic acid. So pyridoxic acid. Then next we will see food sources. Coming to food sources, the vitamin is distributed widely in animal and plant tissues. So it is widely distributed in animal and plant tissues. Rich sources are yeast, rice polishings, then germinal portion of various seeds and cereal grains, and then egg yolk. Moderate amounts are present in liver, kidney, muscle and fish. Milk is a poor source. Highest concentration occurs in royal jelly. Royal jelly is the richest source of vitamin B6. Next we will see metabolic role. Coming to metabolic role, Pyridoxal phosphate acts as a coenzyme. Pyridoxal phosphate. It acts as a coenzyme. It is principally involved with amino acid metabolism. In amino acid metabolism. So first one is, it acts as cotransaminase. Cotransaminase. It acts as a coenzyme for the enzyme transaminases. Act as coenzyme for the enzyme transaminases in transamination reaction. Next one is co-decarboxylases. So it acts as a coenzyme for the enzyme decarboxylase in decarboxylation reaction. Amino acids are decarboxylated to form corresponding amines. For example, tyrosine is decarboxylated to tyramine. Is a corresponding amine of tyrosine plus CO2 by the enzyme decarboxylase. Then coming to glutamic acid, it is converted to gamma aminobutyric acid, then plus carbon dioxide. Then next one is, it acts as coenzyme for deaminase. For deaminase, it catalyzes non-oxidative deamination of these hydroxy amino acids such as serine and threonine. For non-oxidative deamination of hydroxy amino acids such as serine and threonine. Next one is coenzyme for kynureninase. Kynureninase. In tryptophan metabolism, pyridoxal phosphate acts as a coenzyme for the enzyme kynureninase, which converts 3-hydroxykynurenin to 3-hydroxyandranilic acid. 3-hydroxykynurenin to 3-hydroxyandranilic acid. Here also this functions as a coenzyme. Then this ultimately forms nicotinic acid. Thus in B6 deficiency, niacin synthesis from tryptophan does not take place. We will see the formation. Xanthurenic acid. First one is kynurenin. It is converted to xanthurenic acid. This kynurenin can be converted to 3-hydroxykynurenin. 3-hydroxykynurenine. Then it is by the enzyme kynureninase, it is converted to 3-hydroxyandranilic acid. Andranilic acid, it is then converted to niacin or nicotinic acid. Then this 3-hydroxykynurenine can be converted to this xanthurenic acid. Xanthurenic acid is excreted in urine. This is in extrahepatic tissues. This is the formation of xanthurenic acid. In B6 deficiency, kynurenine and 3-hydroxykynurenine, can be converted into xanthurenic acid in extrahepatic tissues, and this xanthurenic acid is excreted in urine. Then next one is transsulfuration. Transsulfuration. It takes part in transsulfuration reactions involving transfer of SH groups. For example, homocysteine plus serine. It gives homoserine plus cysteine. So there is transfer of this SH group. Homocysteine plus serine. It is converted to homoserine plus cysteine. Next one is as coenzyme for desulfhydrases. Desulfhydrases. It catalyzes non-oxidative deamination of cysteine, in which hydrogen sulfide is liberated. That is desulfhydrases. Next one is in interconversion of glycine and serine. Interconversion of glycine and serine. This is by the enzyme serine hydroxymethyl transferase. Serine hydroxymethyl transferase. In this, both FH4 and B6 are required as the coenzyme. FH4 tetrahydrofolate and B6 are required as a coenzyme. Pyridoxal phosphate is required as a coenzyme in the biosynthesis of arachidonic acid from linoleic acid. From linoleic acid to arachidonic acid, there will be pyridoxal phosphate is the coenzyme. Next one is synthesis of sphingomyelin. Sphingomyelin. Pyridoxal phosphate is required as a coenzyme for the activation of serine, which is required for the synthesis of sphingomyelin. For the activation of serine, from this sphingomyelin is formed. Then it is required as a coenzyme for amino acid racemases, then D-glutamic acid to L-glutamic acid, D-alanine to L-alanine. That is amino acid racemases. Then intramitochondrial fatty acid synthesis. Intramitochondrial fatty acid synthesis. It is required as a coenzyme with condensing enzyme for chain elongation of fatty acid in intramitochondrial fatty acid synthesis. Then it is required for active transport of amino acid through cell membrane and intestinal absorption of amino acid. Active transport of amino acid and intestinal absorption of amino acid is required. Then for muscle phosphorylase, it acts as a constant of muscle phosphorylase. Muscle phosphorylase. 4 molecule of pyridoxal phosphate per molecule of the enzyme. So it is a tetramer. 4 molecule of pyridoxal phosphate is required. Then transport of K+. Vitamin B6 promote the transport of K+ across the membrane from exterior to interior. From exterior to interior for the transport of K+. Then as coenzyme for amino acetone synthetase. Amino acetone synthetase. This is required for the formation of amino acetone from acetyl-CoA and glycine. Acetyl-CoA, glycine, amino acetone. So by the enzyme amino acetone synthetase. So in this enzyme also this This is required as a coenzyme. Then synthesis of CoA SH or coenzyme A. Synthesis of CoA SH. Vitamin B6 is involved in the synthesis of CoA from pantothenic acid. In B6 deficiency, CoA level in liver is reduced. Then in porphyrin synthesis, pyridoxal phosphate is required for the conversion of alpha amino beta adipic acid, beta keto adipic acid, alpha amino beta keto adipic acid to delta amino levulinic acid. Here also pyridoxal phosphate is required as a coenzyme. In B6 deficiency, heme synthesis suffers and it leads to anemia. Then next one is hypercholesterolemia. There is a relationship between B6 deficiency, hypercholesterolemia and atherosclerosis. Next one is immune response. In vitamin B6 deficiency, immune response is impaired. Next one is oxaluria. Coming to oxaluria, vitamin B6 deficiency produce oxaluria in experimental animals. These are the metabolic roles. Next we will see deficiency manifestations. Some clinical manifestations are attributed to vitamin B6 deficiency. First one is epileptiform convulsions in infants. Epileptiform convulsions in infants. This has been attributed to pyridoxine deficiency. It is related to lowered activity of glutamic acid decarboxylase. This is for the conversion of glutamic acid to gamma aminobutyric acid. So this is related to lowered activity of glutamic acid decarboxylase, for which pyridoxal phosphate acts as a coenzyme. As a result, there occurs lowering of gamma aminobutyric acid in the brain, which causes this convulsion. Reduced gamma aminobutyric acid, it causes convulsion. Next one is pyridoxine responsive anemia. Pyridoxine responsive anemia. A hypochromatic microcytic anemia called as sideroblastic anemia. It is called as sideroblastic anemia. With high serum iron level and hemosiderosis of liver, spleen and bone marrow may occur with this vitamin B6 deficiency. Pyridoxal phosphate is required as a coenzyme in the reaction by which alpha amino beta keto adipic acid is decarboxylated to delta amino levulinic acid in heme synthesis. So in B6 deficiency, heme synthesis suffers and iron cannot be utilized. Next one is isonicotinic acid hydrazide treatment in tuberculosis. This isonicotinic acid hydrazide treatment in tuberculosis. A syndrome resembling vitamin B6 deficiency is observed in humans during the treatment of tuberculosis. This treatment with the high doses of tuberculostatic drug isonicotinic acid hydrazide. Isonicotinic acid hydrazide. It is otherwise called as isoniazid. Isoniazid or INH. So 2 to 3% of patients receiving conventional doses of isoniazid, that is 2 to 3 mg per kilogram body weight, developed neuritis. Then 40% receiving 20 milligram per kg developed neuropathy. Tryptophan metabolism was also altered, increased xanthurenic acid excretion in urine. Signs and symptoms were alleviated by administration of pyridoxine to these patients. Administration of pyridoxine. 50 mg of pyridoxine per day completely prevents the development of neuritis and neuropathies. It completely prevents. This isoniazid forms a hydrazone complex with the pyridoxine. A hydrazone complex. Isoniazid forms a hydrazone complex with pyridoxine. It is resulting in incomplete activation of the vitamin. Like isoniazid, the drug penicillamine or beta dimethylcysteine, it produces beta B6 deficiency. This drug is used in the treatment of Wilson's disease to chelate copper. It reacts with the pyridoxal phosphate to form inactive thiazolidine derivative. Inactive thiazolidine derivative. Next we will see the therapeutic uses. Coming to therapeutic uses, vitamin B6 is used in the treatment of nausea and vomiting in pregnancy, then muscular dystrophies, treatment of hyperoxaluria, recurring oxalate stones of kidney, then mild forms of pyridoxine deficiency occur in women taking oral contraceptives containing estradiol. A higher intake of this vitamin B6 may decrease the risk of Parkinson's disease. It lowers Parkinson's disease by protecting brain cells from damage caused by the free radicals. These are the therapeutic uses. Next is daily requirement. Daily requirement for adult, it is about 2 mg per day. In infants, it is 0.3 to 0.4 mg per day. Then during second half of pregnancy, it is about 2.5 mg per day. So this is about vitamin B6. This is today's topic. Thank you for watching.